Welcome to my channel World of Forensics. Today we are going to discuss about diatoms, which is a type of trace evidence. Introduction. So what are diatoms? Diatoms are microscopic, unicellular, photosynthetic, eukaryotic algae that inhabit in any water bodies. They may be found in spring, river, pond, lakes, ditches, in any type of fresh water or marine water, and they are also found in terrestrial habitats like wet rocks, moss, soil, any place that are moist in nature in the terrestrial habitats. They have a siliceous skeleton for sure. They may be simple, branched, filamentous, and can be also even enveloped by a gelatinous envelope. Now comes the classification of diatoms. So the diatoms belong to the domain Eukaryota, kingdom Chloromalveolata, phylum Heterocanthophyta, class Basidiophyceae, and order Centralis and Pinnalis. Based on this order, the diatoms are classified into the two types, that is the Centralis and the Pinnalis. The Centralis diatoms are those which have valves exhibiting a radial symmetry, and pinnate diatoms belongs to the order Pinnalis, and they have valve exhibiting bilateral symmetry. Structure and composition of the diatoms. The diatoms are microalgae which are basically present in the water bodies but are also present in moist areas and even in the soil. So the design of the fissures that are present in the diatoms differentiate them from one another, and based on the fissures the diatoms are classified into various species. The cell wall of the diatoms are made up of silica, which is made of silicic acid and is coated with organic layers like pectin, cellulose, and polysaccharides. The diatoms consist of an overlapping halves which cover them, which are known as apitheca and hypotheca. The apitheca and hypotheca are uh, meet at a point that is the griddle and they feed together there. The griddle is the region where both the epitheca and the hypotheca meet, and each theca consists of two parts: the valve, which is the upper flattened top, and the cingulum, cingulum, that is the connecting band which connects the thecas. As we have earlier discussed that diatoms of two types, the pinnalis and the centralis. In pinnalis, there is slit present, and this slit is known as the raphe. If the slit is absent, those diatoms are referred to as pseudopinnalis. The raphe is interrupted in the middle by a thick wall called central nodules. Now, there are rocks which are rock fossils that are made up of diatoms, and this type of rock fossils are known as diatomaceous art or diatomite. These diatoms are also known as the quality chamber in the aquatic bodies. We have a subfield of botany. Diatoms are part of botany, but we have a subfield within it, which is known as forensic limnology. So what is it? It is the field in subfield of botany which examines the presence of diatoms in the crime scene samples and even within the victim, and it also takes uh, takes up the work of examining the diatoms from the victim's body and the crime scene and analyze them and derive various deductions from it, which helps in solving the crimes. Now comes the importance of diatoms in forensics. So diatoms are present in many in the water bodies. So they play an important role in the cases of drowning. So we have two types of cases which are related to aquatic bodies. Either a person goes inside the water when he is or she is alive, and then dies inside the water when such case of drowning occurs, the person inhales diatoms when he or she is present inside the water. When these diatoms enters the respiratory tract, they get into various organs of our body like the lungs, bone marrow, kidneys, brain, and if a person is thrown into the water after he or she is dead, then no such diatoms will be found inside the body of the person, as there is no respiration taking place within that person's body. So the diatoms helps in differentiating the death if it is by submersion or from the immersion of the body. The diatoms also helps in estimation of the postmortem interval or the time since the death. Thank you for watching my videos on diatoms, and this is the first part. In the next part we will discuss about the procedures which are involved in the extraction of the diatoms from the bodies.